during the past couple of days, we have discussed the future of MDS diagnostics and classification and treatment. But now that we have come closer to the end of this Congress, it's necessary to discuss the future MDS Foundation Congress. And for that, I would like to invite Katharina Götze to inform you about the MDS Foundation 2027 Congress.
Thank you, Saskia. So it's my pleasure and my absolute honor to announce that the next MDS meeting in 2027 will be in Germany, in the very beautiful and vibrant and young city of Leipzig. And my co-hosts, Wolf, Carsten, and Uwe, who unfortunately could not be here in Rotterdam, will be very happy to welcome you to Leipzig together with me, and we're very, very excited about this. Just to give you a brief overview for those of you who are not so clear on the geography of, going back, we also have a local committee which will help us and which is composed of a lot of people that have been at this meeting and that I'm sure you're familiar with. So we're very happy to have all of these people on board. And as I mentioned before, if you're not completely familiar with the geography, Leipzig is pretty much in the middle of Germany, a little bit to the eastern part, but from north-south in the middle. It's the gateway to Eastern Europe. It has been the city of peaceful revolution, where before we were reunited from Eastern and Western Germany. And it has quite a reputation in the region for cell and gene therapy. So I think it's a very good choice. And it has good accessibility with a local airport, and it's very close to Berlin with 60 minutes from the direct train. So you can reach it quite easily from all over Europe. This is one of the pictures of the center of town. So it's a historic center that still hasn't been bombed out in World War II, so it's still there and very beautiful. And it's one of the most dynamic and rapidly growing cities in Germany. It has a long history of being a trade city, which dates back to the Roman Empire. And it's home to one of the oldest universities in Europe as well. It's a major center for music, both for classical, so you may have heard of the Gewandhaus in Leipzig, and also for modern. I'm sure Arjan appreciates that. And it's a green city with sustainable growth, and it's internationally renowned for visual and applied arts. So I think it's a very cool location. The venue is a very modern Congress center. It's bright, it's modern, it's well equipped for this meeting. So we really hope you will join us. We hope we will have an exciting and innovative meeting. But I have to say, Saskia and Arjan have really upped the bar, and it's going to be very hard to beat this wonderful meeting that we've had here, fabulous meeting in Rotterdam. I've really enjoyed it. Nevertheless, we're excited to host the meeting in Germany. The last time was 2013 in Berlin, and we'd love to welcome you all. Please save the date, and thanks.
So now we're going to proceed with our award ceremony. So I would like to invite Stephen Nimer to join us on the stage.
Okay, good afternoon. It's my great honor to introduce one of the most important recognitions in our field, the Young Investigator Grant Award. The award not only recognizes important research, but is a deliberate investment in the future of MDS research and patient care. We've had an incredible meeting, and if you've stayed awake during even one of the talks, you'll know that MDS is both a complex and a challenging disease. Urgent unmet needs in diagnosis, treatment, and patient quality of life. And I must say, Guillermo, one of the most inspiring talks was yours really presenting so many options and the future, and many of the talks doing the same. So as some of us in the audience get older, it's very important that we invest in our future. And so the MDS Foundation has been presenting Young Investigator Grant Awards over the years, and today we acknowledge two very talented individuals. One is here with us, the other is in California, and I think the first recipient of the award is Xin-Li Lu, MD, PhD at Stanford. Sydney's talk is on novel mechanisms of oncogenesis for SRSF2 mutations. He receives funding for this project, and Sydney is an MD, PhD who did training, got his PhD at Weill Cornell, did his residency there, did a fellowship at Sloan Kettering, and is now an assistant professor at Stanford. And so congratulations to Dr. Xin-Li Lu, and I think he's going to be joining us via Zoom and giving his talk.
So thank you so much for this great honor, and I apologize that I can't be there in person. Let me start sharing my screen, and hopefully this is visible to everyone. So yes, I'll be talking about 3' information in SRSF2 mutant MDS for the next 10 minutes or so. Let me go ahead here. I have no disclosures and no conflicts of interest. This audience will not need any introduction to recurrent mutations of SRSF2, which were discovered in 2011 in MDS and in related disorders. And for my work and for what I'll be talking about today is the impact of this mutation on 3' information in MDS. So this audience is, of course, very familiar that hotspot mutations of SRSF2 of proline 95 cause a host of RNA splicing alterations, which are known to be important for disease pathogenesis. But what we've been wondering is whether or not they also change 3' information, which is also known as cleavage and polyadenylation, in addition to the splicing changes, and whether these could be important. So just to get everyone started on the same page, this process of alternative cleavage and polyadenylation, or 3' information, I have on the screen here a cartoon of a immature messenger RNA, which has protein coding exons and a 3' untranslated region. And very broadly speaking, and I should mention there are different places along this immature messenger RNA where the transcript can be cleaved and a poly A tail added for things like translation. And very broadly speaking, there's two different kinds of alternative 3' information that can happen. If we cleave at different sites inside the 3' untranslated region, we get transcripts which have the same protein coding exons, and so which encode the same protein, but which differ in their 3' UTRs. And this can lead to very important alterations of cellular function, with, for example, CD47 being a major example. The other change is even more drastic, which is that it's possible to cleave and polyadenylate transcripts before decoding exons have finished being encoded. And in that case, the transcript encodes a truncated protein, which can obviously have different biological effects. In the world of hematology in general, normal 3' information is actually quite important. It's known to cause forms of thalassemia, and it is a mutation in the cleavage site is in fact the cause of prothrombin G2210 thrombophilia, which is of course a very common hypercoagulable syndrome. In malignant hematology, there's been a lot of formative work done by Dr. Christine Mayer, who's now at Memorial Sloan Kettering, showing that 3' UTR shortening can activate oncogenes. and that this upstream process, which causes truncated proteins that I was describing to you earlier, can inactivate tumor suppressors. And while it's really been studied quite a bit in solid tumors, really the only work in myeloid disease that I'm aware of is some work by Dr. Dong'er Zhang's group at UCSD, where they knocked down a core component of the 3' end formation complex and showed that this was therapeutic for core binding factor AML. So our major hypothesis for this work is that oncogenic SRSF2 mutations can alter this cleavage and polyadenylation process in MDS, and that this might regulate disease biology or present novel therapeutic targets. And one might ask, okay, but why might SRSF2 regulate cleavage and polyadenylation? It turns out that there's been a number of publications now on a very closely related protein, SRSF3, which is known to actually regulate cleavage and polyadenylation, and also a paper that shows SRSF7 modulating this process. And in fact, they both biochemically interact with the cleavage and polyadenylation complex. There's even more recent unpublished work from Dr. L. Sterling Churchman's group showing that SRSF1 also regulates cleavage and polyadenylation. So armed with that hypothesis, of course, we wanted to go ahead and see whether or not indeed the mutant SRSF2 does that. And the easiest thing that we might do would be, of course, to look at all the published RNA sequencing data out there that exists for both patient samples as well as isogenic cell lines and other models. However, it turns out that to study this kind of RNA processing, we can't really use RNA-seq. And so what I have on the screen here is a cartoon of a metagene plot that essentially shows read coverage in next-generation sequencing from the beginning of the transcript to the end, basically with the point that RNA-seq really lacks coverage and even coverage at the 3' ends and actually covers the entire transcript, which is problematic, believe it or not, for accurately defining these ends. And so over the past few years, we've set up 3' end sequencing, which is a technology that specifically looks at the 3' ends of transcripts that allowed us to answer some of these questions. This is what our data looks like. Here in the left panel, what I have for you are changes to lengths of 3' untranslated regions. So just to remind you, these are genes where the alternative cleavage and polyadenylation don't change the protein coding sequence, but change the 3' UTRs. And here in the blue are dots, and each dot here is a gene of genes whose 3' UTRs are lengthened in cells with an SRSF2 mutation. And I neglected to mention that these are pilot studies of MDS patient specimens that I'm showing you here. And the red dots, in this case, there's more genes that have actually 3' UTR shortening in SRSF2 mutant MDS. On the right-hand side of this plot here, what I have are changes in this other form of polyadenylation that I showed earlier in cartoon form that causes truncation of the protein coding sequence. And by changing the truncation, either, yes.
Oh. There seems to be a problem with the slides here in the audience, in the auditorium. Okay. Now we see a slide entitled RNA-seq.
Oh, we're having some synchronization issues then.
Yeah.
I'm showing a slide that has red and blue dots. I'm not sure if that's coming up.
Right now we see a slide that says Rotterdam 2025. So we are not showing any data, and I don't know if we can, one of your slides just flashed on and then flashed off. So I don't know if the AV person can pull these slides back, the PowerPoint. There we go. Now it's, yeah. SRSF2 mutant MDS exhibits 3' UTR and upstream alterations. And we see panel A.
Okay, great.
And now we see some blue dots. Yeah.
Is panel A showing up?
Yes.
Okay, I apologize for the technical glitch, and I'll sort of skip a little bit quickly. And so we can see essentially genes with 3' UTR lengthening that are driven by mutations of SRSF2 in MDS patients, as well as genes with 3' UTR shortening. And then in the protein coding sequence, because of upstream polyadenylation, we can also see both genes that have increased upstream polyadenylation, so more truncation of the protein coding transcript, as well as those with less truncation, so more full-length transcripts. Coming to one particular gene that I would like to end the talk on is the gene BRD4, bromodomain containing 4. And I'll first start here at the bottom half of the slide, where what I have are gene diagrams for the C-terminal half of bromodomain 4. And BRD4, we now understand, has two transcript isoforms. There's a full-length or long isoform, which has many protein coding exons. And then there's a truncated or short isoform from this upstream polyadenylation process, which has a shorter 3' UTR. And here in panel A, what I have for you is this 3' seq data that I have been describing, that we've been generating. And so here shaded now is a polyadenylation site corresponding to the generation of the short isoform, indicating by the height of the peaks that there is increases in SRSF2 mutant disease. Whereas here in the purple shade, there's much less difference in the polyadenylation of the full-length isoform. In this case, we also see correlates by the amount of coverage in RNA-seq, and also by nanopore full-length read sequencing support of this observation. Just to say a little bit more about why we are choosing to study BRD4, as I mentioned, there's a full-length isoform that has bromodomains in extra-terminal domain and a C-terminal domain, and then a short isoform which lacks significant parts of the C-terminal domain. It's now understood, and this has really been studied only over the past few years, the differences between long and short is that BRD4 short is, in a word, pro-oncogenic in a number of solid tumors, that it inhibits DNA damage or repair, and that, of course, in the sister disease, AML, that suppression of this isoform can actually slow disease growth. On the other hand, we are now starting to understand that the full-length BRD4 long can actually be tumor suppressive in some solid tumors, although its role in hematologic malignancies is still being, of course, studied. And in this case, there's some manuscripts that show that it actually promotes DNA damage repair. This is my last data slide. I will first say that the previous data that I showed in the dot plots and in the sequencing coverage plots was in patients, but we were also able, in a collaboration with Dr. Andy Reinisch, who's at the University of Graz, to actually use isogenic cells, so K562 cells, THP1 cells, and others, which are wild type or SRSF2 mutant. And we can, again, in these isogenic cells, very clean systems, see by qPCR upregulation specifically of the BRD4 short isoform with much fewer changes in the long isoform. This here in panel B is 3' rapid amplification of cDNA-N, so a RT-PCR based confirmatory method, which shows the same thing in these cell lines. And finally, this is why I think we're excited to go after this target, which is that alterations in the transcripts of BRD4 short actually are reflected by Western blot in increased levels of the BRD4 short protein in SRSF2 mutant isogenic cell lines. So the conclusions of our work is that we've identified a number of mutant SRSF2-driven transcript 3' end changes in MDS patients and cell lines. I didn't get into this very much. The targets are actually quite distinct from those with splicing alterations, as we think of them, things like EZH2, caspase-8. And we're continuing to study MDS patients and BRD4 isoforms to understand their role in biology and to target them therapeutically. I'd like to thank the lab. This work that I showed you was primarily done by Agata Sakreka, a very talented postdoc, together with Shunghong Cao. Our collaborators, I mentioned Andy Reinisch at the University of Graz. Dr. Artisha Singh really is a computational maven who helps us with a lot of the 3' end computational analysis, and would like to gratefully acknowledge the generous support of the MDS Foundation and the other funding sources. So thank you so much for your attention, and happy to take your questions.
Okay, well, I think, thank you so much, and good luck with your project, and we look forward to hearing you present your work at one of our future symposium. I'm also thrilled to award Dr. Lin-Pierre Zhao, MD, PhD. Lin-Pierre is an assistant clinical professor and a full senior physician at the hematology unit at the Hôpital Saint-Louis in Paris. And his project is entitled Unraveling the Immune Impact of Clonal Hematopoiesis and Myelodysplastic Syndromes through their interaction with immune-mediated inflammatory disorders. And Lin-Pierre is here, and I'm going to present him in 1 second with the award, and then he'll begin his talk. So let's congratulate him as well. Congratulations.
Good afternoon, everybody. Let me just start by saying it's a great honor. And first, I'd like to thank the Foundation for supporting this project. And I'm very grateful, and thank you again for letting us present this project. So I'm a physician at the St. Louis Hospital in Paris, and I will present our research project on the interplay between clonal hematopoiesis and immune-mediated inflammatory disorders. That is a project that aims to explore how clonal hematopoiesis contributes to systemic immune dysregulations. And I have no conflict of interest. So immune-mediated inflammatory disorders, or IMIDs, affect around 10 to 15% of patients with MDS. And these include a heterogeneous but yet well-defined group of conditions, including inflammatory arthritis, connective tissue diseases, systemic vasculitis, all with inflammation at the systemic level. And when they are associated with MDS, these IMIDs are often clinically atypical in their presentation and display a high rate of refractoriness to standard immunosuppression. So one turning point in the field has been the discovery of VEXAS syndrome. That is a paradigmatic example of how a shared genetic mutation here in UBA1 gene is driving both the hematologic and the inflammatory phenotype. And beyond VEXAS, we have reported that MDS patients with IMIDs are enriched for some specific mutations, notably in epigenetic regulators. And we and others have also observed that IDH-mutated patients display a specific inflammatory phenotype, mainly with articular and cutaneous involvement. These are unpublished data. And that targeting the mutated clone, for example, using IDH inhibitors in IDH-mutated patients, or azacitidine in VEXAS patients, this has been also published by the Dutch group, I think, can lead to both hematologic and inflammatory remissions. So altogether, this supports the idea that the clonal architecture and composition may shape the inflammatory manifestation at the systemic level. But while the association between MDS and IMIDs is increasingly well recognized, much less is understood at the level of clonal hematopoiesis. We know that CH has been associated with inflammatory complications in non-hematologic context, such as cardiovascular disease, COPD, liver fibrosis. But the mechanism, especially driving this in the adaptive immune compartment, remain poorly understood. So our central hypothesis is that CH contributes directly to the development and the persistence of IMiDs. And to explore this, our project will combine epidemiologic, experimental, and translational approaches to define the immune consequences of CH. So this is a very early stage project, so I do not have any results to show you, hopefully more in 2 years. Nevertheless, the work is organized around 3 main objectives. First, we'll examine the association between CH and IMiDs in the general population. Second, we will model CH and IMiDs in mice, specifically through a model of inflammatory arthritis. And third, we'll investigate the immune mechanisms underlying these interactions, focusing on how CH alters the function of both innate and adaptive immune compartments. So in AIM 1, we will use exome sequencing data from participants of the UK Biobank, and IMiDs will be classified based on ICD-10 codes. We'll assess the incidence of IMiDs in CH carriers versus non-carriers, adjusting for classical risk factors. We also plan to examine whether certain mutations are more correlated with specific IMiD subtypes. And in parallel, we will investigate whether the presence of IMiDs increases, in turn, the risk of CH progression using time-dependent statistical models. In AIM2, we turn to in vivo models. We've established tattoo knockdown bone marrow chimeras in irradiated mice. And we will first use rheumatoid arthritis models because RA is the main IMiD associated with MDS, but we are currently setting up other IMiD models. So we will use, without going into further detail, 2 arthritis models that will allow us to explore differentially the impact on innate and adaptive immune compartments. So our preliminary data confirm that we are able to induce arthritis and suggest early signs of increased inflammation and bone erosion in CH mice. And we will use as readouts clinical scoring, histology, and micro-CT scan to quantify the joint damage. Importantly, we will also test in these murine models the impact of therapies, notably azacitidine, but also emerging therapies in the context of CH, like vitamin C and canakinumab. And this will help us determine whether targeting the clonal compartment can mitigate inflammatory responses. And finally, AIM3 focuses on the immune mechanisms that underlie CH-associated inflammation. So we'll first use site-seq to profile immune cells in the murine models. We'll validate these findings using at least some targeted functional assays that will include transrail migration assays to test the cytokine-induced immune cell trafficking, antigen-specific responses in T and B cell compartments, and also reconstitution experiments to isolate specific cell type contribution. Finally, we try to translate these findings into the clinic. So we've already assembled a cohort of ERA patients with matched controls with both blood and synovial, the articular liquid samples. And we'll perform flow cytometry and targeted sequencing. And we expect that CH will associate with more severe disease, including local infiltration by clonal cells. So we hope this project may significantly advance our understanding of how CH may be a driver of systemic inflammation by reframing CH as a driver of IMiDs. And we anticipate the initial results from the epidemiologic analysis and murine models within the first year, followed by in vivo and translational findings over the following years. So I'd like to thank again the MDSF Foundation for supporting this project. I'm very grateful for the opportunity to lead it. I'd like to thank also my mentors, Pierre Fenot, Lionel Alès, and Marie Sebert, who are in this room, my collaborators and teams at the Leukemia Institute Paris Saint-Louis. And thank you for your attention.
Thank you very much, Dr. Zhao. And Dr. Zhao and Dr. Lu, we look forward to seeing your impact on the field over the next decades. Thank you so much and congratulations. And now I'd like to turn things over to Arjan and to Saskia to give out some additional awards.
Yes, we would like to award the Tito Bastianello Awards. And Tito Bastianello was born in Venice in Italy in August 1930. And after graduating in law, he started working in a family business until 1958, when he established his own company and one of the first chains of supermarkets in Italy. And he was very much engaged in social, cultural, and also non-profit activities. But during his lifetime, he was also challenged with difficulties, both in his business and in personal life. And in 2001, he was diagnosed with MDS. And at that time, there were no drugs which could counter this disease. And he put an enormous effort in looking at the possibilities of treatment. And when in 2004, the effectiveness of azacitidine was confirmed, he went to the US and then to Germany in the hope of being treated with azacitidine or decitabine. But those attempts were initially unsuccessful. In the end, he could unfortunately not beat the disease and knew that his battle was lost. He did get treatment with new drugs, but unfortunately, as I said, he lost this battle. But thanks to, he knew that participation in clinical trials could give hope to new MDS patients in the future, even though it was ineffective for him and he passed away in September 2005. And the Tito Bastianello Awards in his remembrance are awarded to the best abstracts of this conference.
So we are so happy that the review committee selected 3 prizes this year. And please, Dr. Sezaki, take the floor. And we are happy to give the first award to you. And actually, Dr. Sezaki received her Master of Science in Molecular Medicine in Germany, in Göttingen, after which she continued her research in Japan. And later on, she moved to USA, where she is now a fellow, postdoctoral fellow in San Antonio. And we are very grateful that you can receive the award today from the 18th International MDS Foundation Review Committee. Congratulations.
So thank you for the kind introduction. I'd like to thank the MDS Foundation and the committee for selecting our abstract to present today, and of course this award. So to start with a bit of context, as we have heard from many experts so far, managing low-risk MDS anemia can be a challenge, as many of these patients will eventually experience resistance to treatment. And despite efforts to address this issue, there aren't that many good low-risk MDS models out there that can sufficiently reproduce MDS phenotypes. So we tried to address some of these issues, bring a little bit of clarity, which I have the pleasure of sharing today. So in our bioinformatics analysis of primary MDS samples, we noticed that around half of MDS patients have high TGF-beta signaling, and that these patients also show downregulation of TCA cycle enzymes, including mitochondrial complex II, also known as succinate dehydrogenase, or SDH. SDH is an enzyme complex that participates in both oxidative phosphorylation and the TCA cycle that converts succinate to fumarate. So as a functional link between these two fundamental processes, SDH dysfunction has been associated with a broad range of diseases. And to first test whether SDH downregulation is a direct effect of TGF-beta, we treated human bone marrow CD34 positive cells with TGF-beta 1, collected the cells after 48 hours, extracted the mitochondria, and indeed, we were able to confirm reduced SDH expression. Based on these results, we next wondered whether the knockdown of SDH alone, and in the absence of any somatic MDS driver mutations like TET2 or DNMT3, is sufficient to reproduce MDS phenotypes. We generated a transgenic mouse model that is doxycycline inducible and has blood-specific knockdown of the SDH subunits A, B, and D using short hairpin RNAs and by crossing with VAV1 mice. These mice express reduced SDH subunits while the assembling factors are unaffected. We also saw an increase in HIF1A expression, which aligns with our previous work, where we found that if pseudohypoxia state caused by HIF activation underlies MDS phenotypes, largely due to the accumulation of oncometabolites like fumarate and succinate that can block PHDs and stabilize HIF. We first phenotypically characterized these mice. We found that knockdown of SDH in blood leads to hallmark features of MDS, such as cytopenias and anemia. We saw a decrease in white blood cells, red blood cells, reduced levels of hemoglobin and hematocrit, while the MCV, which is the average size of red blood cells, and RDW, which measures their size variation, increases over time. This is well reflected in peripheral blood smears, where microcytes, macrocytes, and teardrop cells indicate unequal RBC size. We also saw dysplasia in 3 major blood lineages in bone marrow smears, characterized by hypersegmentation, binucleation, and micromegakaryocytes, among other anomalies. So phenotypically, the SDH knockdown model presents hallmark features of low-risk MDS. These mice also show phenotypes of refractory anemia with ring sideroblasts, which are erythroblasts that accumulate iron in their mitochondria and have been a challenge to reproduce in some of the other MDS models. So to try to understand the cause of anemia in these mice, we next analyzed the erythroid progenitors. Within the bone marrow, we see sinusoids that are filled with TER119 positive cells in the control, while in the knockdown, these sinusoids are practically empty. In the spleen, the majority of the TER119 positive cells found within the red pulp are enucleated and mature, while in the knockdown, these cells are still nucleated and immature. From our flow analysis, we noticed a significant decrease in the most mature stage 5 subset of the RBCs and an increase in the intermediate stage 4, both in the bone marrow and the spleen, suggesting a maturation block that results in overall reduction of terminal RBCs. We next investigated how SDH downregulation affects the stem cells leading to these phenotypes. We performed single-cell RNA-seq on bone marrow HSPCs, induced short-term for 2 weeks to see if there were any transcriptional changes. We first checked reduced expression of the SDH subunits in quiescent and activated HSCs to confirm that our model is working. The differentially expressed genes that were significantly downregulated involve those related to HSC fitness and metabolism, like CDK8 and ApoE in quiescent HSCs, while cell cycle-related pathways were upregulated in activated HSCs, indicating proliferation and differentiation. We next checked the stem cell compartment after 8 weeks of long-term induction, when MDS phenotypes become markedly evident. There was a significant expansion of the MPP2 subset and a near-complete depletion of the stem cells. We also noticed a significant decline in total bone marrow cell number, indicating a bone marrow failure phenotype, largely due to the increase of apoptosis, which has been reported in low-risk MDS patients. We next evaluated the functionality of these stem cells with serial colony plating and comparative bone marrow transplantation. Results from both experiments were inferior to control bone marrow cells, indicating the defective nature of these stem cells. This model is, however, reversible. When these mice are taken off dox and SDH function is restored, the blood parameters were able to recover within 2 weeks. With this model, we were able to show that SDH downregulation alone, and in the absence of any MDS-related mutations, is sufficient to recapitulate low-risk MDS phenotypes. In other words, these phenotypes are largely metabolically driven. Our initial starting point was that TGF-beta high MDS patients show downregulation of SDH genes. Using an array-based assay, we checked the bone marrow supernatant of control and knockdown mouse and found increased TGF-beta1 among other inflammatory factors. So as TGF-beta1 is increased, we next tested these mice using Luspatercept, which works by trapping TGF-beta superfamily ligands and preventing downstream SMAD signaling. And our mice responded favorably to Luspatercept, showing recovery of major CBC parameters marked in blue, whereas treatment with EPO in red did not. Flow analysis confirmed that Luspatercept treatment results in a boost of stage 5 mature RBCs. And furthermore, we noticed that the stem cell compartments can also be rescued. Now, with the model at hand, we next investigated the mechanism of Luspatercept action. We performed single-cell RNA-seq of KIT-positive cells from untreated and treated mice and found 2 populations that showed a striking change in gene expression. These were the erythroid progenitors and a subset of bone marrow macrophages. First, we confirmed that Luspatercept treatment does not restore SDH expression. The SDH subunits B and D remain downregulated, similar to the knockdown, allowing us to rule out the possibility that our model was simply reversed. And from our pathway analysis, we found that Luspatercept works through several mechanisms on erythroid progenitors. First is that they enhance a compensatory mechanism to upregulate other TCA cycle enzymes and mitochondrial complexes besides SDH, like IDH2 and complexes 1 and 4. Next, they upregulate serine glycine biosynthetic processes, which is important for downstream heme synthesis and hemoglobin production. We also noticed several prominent regulators of erythroid proliferation and differentiation and hemoglobin synthesis. The other population that expanded post-treatment were these macrophages located as distinct cluster away from bone marrow-derived monocytes. They are characterized by genes specific to erythroblastic island macrophages like SPICE, CD169, and MERTK, which are important for erythroid differentiation, maturation, and nuclei engulfment. So erythropoiesis occurs at EBIs, where a central macrophage serving as a supportive nurse cell provides growth factors to surrounding erythroid progenitors for RBC maturation. In these macrophages, phagocytic and endocytic pathways are upregulated, which could suggest their increased ability to nucleate terminal erythroid progenitors. Also, some iron recycling genes, like transferrin and ferroportin, are also upregulated, which means more efficient clearance of iron, the recycling, and incorporation into heme. This can be seen in spleen sections that show reduced iron buildup after Luspatercept treatment. We next imaged the bone marrow to see whether these EBI-related changes could be visualized. As the SDH knockdown mice expressed GFP upon indox induction, we are able to see an increase in these GFP-positive cells that are large and foamy after Luspatercept treatment, while these high ploidy CD41-positive megakaryocytes are largely displaced, possibly due to an altered bone marrow microenvironment. These foamy cells are often found adjacent to the sinusoids and are in contact with many TER1-9-positive cells. They also express typical EBI macrophage markers like CD169 and TIM4. After Luspatercept treatment, these cells associate significantly more with red blood cells. They also appear to help with their nucleation, indicated by the increased ratio of enucleated to nucleated cells that they are in contact with. In summary, we found that metabolic dysregulation through SDH can recapitulate high TGF-beta MDS phenotypes. It is known that TGF-beta signaling inhibits late-stage erythropoiesis, and Luspatercept can counteract this. Using the SDH knockdown model, we uncovered previously unknown mechanisms of Luspatercept MOA through cell intrinsic and extrinsic means. Luspatercept can act beyond late-stage erythropoiesis by maintaining the energy demands of erythroid progenitors and upregulating RBC maturation factors, as well as by expanding a subset of macrophages for EBI assembly, iron recycling, and efficient erythropoiesis. Interestingly, we noticed that roughly 30% of SDH knockdown mice do not respond to Luspatercept, while around 60% of mice were responders that showed either a sustained or a greater than 10% increase of CBC parameters, a situation that is similar to the primary resistance in some of MDS patients. When we analyze the bone marrow erythroid cells in responders versus non-responders, the non-responders still have erythroid progenitors, and the difference lies mainly in the terminal maturation stage 5. The problem isn't cell intrinsic. So we imaged femur bones from responders and non-responders, the non-responder bone being completely white with only patches of red at the metaphysis. Compared to a pretty empty white bone marrow, active erythropoiesis could only be seen where GFP-positive cells were present. So we think that the underlying reason for a lack of Luspatercept response could be due to an exhaustion of these EBI macrophages, which really depends on the patient's inflammation profile. And as a final note, also reported from the Medalist trial was a rescue of neutrophils in low-risk MDS patients treated with Luspatercept. We similarly noticed an increase in the cell frequency of all neutrophil progenitors in Luspatercept-treated bone marrow. We found DEGs enriched for pathways related to neutrophil activation, differentiation, and upregulation of translation, indicating a cell-intrinsic response similar to erythroid progenitors. We also found a positive correlation between RBC and neutrophil fold change, so maybe the similar energy demands between erythroid progenitors and neutrophils, both being highly dependent on OXPHOS, underlie the Luspatercept rescue. So in summary, TGF-beta high MDS patients show downregulation of SDH, and SDH knockdown recapitulates low-risk MDS phenotypes, but importantly, the MOA of Luspatercept is through metabolic adaptation of ERPs and neutrophils and assembly of EBI macrophages, and non-responders and secondary resistance could result from an exhaustion of these EBI macrophages. So here are my acknowledgements. I'd like to thank all members of the Gang Huang Lab and our collaborators, and thank you very much for your attention.
Thank you so much, and it's quite clear that you have really a prize winner for this excellent work. Thank you.
Thank you so much.
The next Tito Bastianello Award will be awarded to Luca Janssen. And Luca obtained her medical degree in 2019 and afterwards started her PhD project at the hematology department of the Amsterdam University Medical Hospital. And her research focuses on immune profiling in myelodysplastic syndromes. And her abstract is titled Immunophenotyping Typical Predictors of Lenalidomide Response in Lower Risk Myelodysplastic Neoplasms, the HOVON 89 trial. And before you start with your talk, we would like to present you with the certificate.
Van harte.
Well, hello everyone. My name is Luca, and I'm in my final stages of my PhD project at a group of Arjan. And today, on behalf of our research collaboration with researchers from the Radboud UMC, I am excited to share you some data of our immune profiling studies on the HOVON 89. And the HOVON 89 trial, patients were included in low or intermediate risk at the time determined by the IPSS. And all patients received lenalidomide with or without erythropoietin-stimulating agents. And erythroid recovery was the response, was how we defined response to lenalidomide. And we saw that, of course, in del(5q patients, we observed lenalidomide response, but also in non-del(5q patients, the response was observed, albeit at a bit of a lower rate. And in addition, also flow cytometric data, cytogenetic data, and molecular data that were already collected at the study. Also biomarkers that could predict this response to lenalidomide were investigated. And we already found that besides the presence of a deletion in the 5q, also the presence of lymphocytes and progenitor B cells in the bone marrow were in favor of response to lenalidomide. And in contrast, the number of mutations or the presence of an SFTP1 mutation was negatively associated with lenalidomide response. And this was published last year in Leukemia. And with this in mind, we were thinking, like, we know that the presence of lymphocytes in the bone marrow are associated with this response, but we didn't really know which type of lymphocytes and also what other types of immune cells are present and could be involved in this effect of lenalidomide, especially in these non-del(5q patients. So we included frozen bone marrow samples from 49 patients within this cohort, of which 25 were responders and 24 non-responders. And we added 11 HMS hematologically healthy donors. And we performed a comprehensive flow cytometric panel, including a lot of markers to be able to distinguish progenitor and stem cells, as well as a lot of various of immune cells and functional markers. And in the end, we were trying to see if we can find select features, immune features that are able or that are associated with lenalidomide response. And we pre-gated or we gated, by experts gated all the immune cells and immune markers, and we merged all these immune features in one master file, and we had a limited amount of patients, so a lot of features, and we performed first, we tried to perform a univariate analysis to see if we can find features that associate with this response, but corrected for multiple testing, this was not really helpful in our analysis, which is common in high-dimensional analysis. So what we did was actually a computational approach, and we decided to use a lasso regression model, and the first step in this computational approach was to optimize this model by running the model 49 times, so as many as patients that we have. And first we had a part of them were training sets, and one patient was in a test, and through this process we were able to optimize our lasso regression model by the so-called lambda. And with this optimized lambda, we were able to identify a selection of features that combined could detect our lenalidomide responders. And in our model, so here you can see that some have actually a positive correlation with response and a positive coefficient, and others have a more negative coefficient with response. And it's actually a combination of these features that is in the end able to predict lenalidomide response. And of course, we were also interested in the more biological aspects. So of course, you can make box plots of these features as well to see what are the actual percentages of these markers and also the expression levels. And interestingly, we were able to capture this combination of these immune features in this principal component analysis. And there you see the combination of these markers are all able to contribute to distinguishing these patients. And what you can appreciate is that we see that there's actually sort of a gradient from the left bottom to top right, where in blue you can see the responders in the left bottom of the figure, and you see the non-responders in red on the right upper corner. So there's sort of a diagonal imaginary line. You can imagine that there's actually a distinction between these groups. And what we did next was we wanted to see some additional patient characteristics that we have of these patients, such as a del(5q), which we know, of course, that patients with a del(5q) have a higher response rate. So we were interested in how this related to our identified immune features. And here you can see that actually the del(5q) patients, there were 3 from 11 in total that we have in our cohort that had a del(5q), are actually in the non-response part of from the graph, in addition, we also did the same for NSF3B1, which is correlated with the non-response. And here you can see that it is actually more randomly distributed across the PCA. And from these two figures, we actually have the hypothesis that our immune-phenotypic associations with lenalidomide response seem irrespective of a del(5q and NSF3B1, and maybe not disease-specific, but maybe really relevant for lenalidomide response. In addition, we also try to see how this resembles to our healthy donors. So we have our PCA of our patients, and we overlaid the donors on this PCA. And what you can see that in green are the healthy donors, and they actually are located in the part of the graph in the gradient where the responders are located. So the combination as well in the responders are more similar to controls. And also what I wanted to show you is one of the last figures for now, is the correlation of our 9 immune features with actually all the other features that were present in our panel. And we performed Spearman correlations and selected those that had a correlation above 0.6 or below, negative below 0.6, which indicates a moderate or a strong correlation. What I wanted to highlight here is that if you look into not only the presence of certain cell types, it's actually the marker expressions that are highlighted. So, for example, the TIGIT expression is also expressed on other cell subsets, and also the KLRG1 or the OX40 or the CD80 on non-classical monocytes. You can also see that these are also correlated with the expression of this marker on other myeloid subsets, which may indicate that there's actually more going on. It's not only a sort of subset of cell type that is interesting to take into account, but there's more of a response or immune status in this bone marrow. So in short, what I've shown you is that we have identified a couple of immune features that in combination is able to distinguish lenalidomide responders. And this seems irrespective of a del(5q or an SF3B1 mutation. And also, it seems that these are more like healthy donors. And a mirrored effect can be seen for certain marker expressions. And what is important from our study, I think, is that our data indicate that the immune phenotype is actually quite interesting to investigate, of course, in response prediction. It can also give you insights in new targets for therapy. And of course, it's important in our current era of precision medicine to also take this immune phenotype into account. And with that, I would like to thank a lot of people that have been involved in this work, especially my supervisors, Arjan and Marisa, also Caroline and Diede, mostly for the foundation of all the immune profiling that we have performed, Tim for all the statistical and help that we have, I've had, and also Willemijn and Joop for their involvement in the study. And now I'd be happy to answer any questions if you have. Thank you.
Thank you, Luca. That was fantastic work and a well-deserved award. So thank you very much.
So then we have the final awardee, and I would like to introduce Dr. Prummel, and she is over my right. Please come on the stage. And she is actually a postdoctoral researcher at the moment in Heidelberg, and actually she did her PhD in Zurich in Switzerland. And her talk will also on immune cells in the microenvironment especially, and we're looking forward to your talk. However, first we would like to congratulate you to having awarded. Congratulations for that.
Yeah, thanks a lot for this kind introduction and also for the recognition of my work. It's quite an honor to be selected for this award, and I'm excited to share my key results with you today that I have been doing during my postdoctoral time with Judith Zauke at the EMBL Heidelberg. I have no conflicts of interest. So first, I want to recap a little bit about the bone marrow microenvironment. So this is in the core for what I will do during my postdoctoral work. And we see this as the multitasking ecosystem. So central, of course, in the bone marrow is hematopoiesis. And this hematopoietic differentiation happens due to an interplay between cell intrinsic but also cell extrinsic factors, which come from the bone niche microenvironment. So these are cell types derived from MSCs, but of course also of so mesenchymal stromal cells, but also of course from the endothelium. And of course also on the other angle there is the immune system there as well. And what we also, what we basically study, or what is my main focus, is to understand how the stem cell niche is disrupted in the evolution of bone marrow malignancies, but also in the context of aging. And to talk about aging, and this was also already earlier in this meeting introduced by Suzanne Winter with a really nice introduction, is that actually the hematopoietic system and the niche are changing and disrupted with aging, actually. And one of those elements is that we actually see the diverse output of the mesenchymal stem cells in the bone marrow, so there is an increased adipogenesis. On top, in hematopoietic system, we see a myeloid bias, and of course there is an increased prevalence of mutated HSCs that have a proliferative advantage in the bone marrow. So then we talk about actually clonal hematopoiesis. And most of these clonal hematopoiesis is driven by mutations in epigenetic regulators, for example, DNOT3A or TAT2, or sometimes in spliceosome genes. So in this project, we were very curious to understand how the bone marrow niches were modeled in CHIP individuals and in low-risk MDS patients. And we focused here also then on MDS with spliceosome mutants like SF3B1 and SRF2. And yeah, we were particularly curious, like how does this niche actually shift from a healthy towards a CHIP and MDS supporting state, if this happens, and then which are the stromal cells actually contribute to support this mutant and neoplastic states, and how the immune system has a role in remodeling this niche. And particularly, a lot is still missing about the human context and also on a spatial and single cell level. So this project was a tight collaboration with Boring Gasgas, who is at the University of Mainz and also part of the CHOICE consortium, where we already have heard about more in this meeting. And we made use of the Bohemi cohort, also from Erythroblastic, which is a really nice cohort of lots of patients that have mutational profiling, have biobanked peripheral blood, bone marrow aspirates, and especially also FFPE tissue blocks, so bone marrow biopsies, which are very of interest for us as well. And we performed multiple molecular assays in the following cohort. So we had various healthy non-CHIP donors included, 16 CHIP patients in total, and 32 low-risk MDS. Yeah, so just to tell you about like our design, so we have had an angle on the imaging part, so we performed immunohistochemistry on the bone marrow biopsies, and then we performed transcriptomics. So we did both single-cell transcriptomics and targeted RNA-seq. And first I want to share a bit with you about our targeted RNA-seq experiments. So this we did using nanostring predefined panels, which are the immune exhaustion and immune panels. So when we perform principal component analysis, we see a nice clear separation of the MDS, SF3B1 mutant MDS, and distinct from the clonal hematopoiesis and controls. And actually you do see that there is also even two groups on the healthy and CHIP donors in the PCA space. I'm not going to share so much more about it, but there are some reasons why they actually separate on the PCA. And then when we performed differential expression analysis, we did see significant genes up and down regulated compared to the healthy controls, and even already in CHIP, although with a less extensive fold change, but we see similar genes actually changing also if we compare whole bone marrow controls versus CHIP. And when we do a gene set enrichment analysis, we see that, for example, TNF-alpha response to cytokine and inflammatory responses are terms that pop up. And of course, we did this targeted RNA-seq on bulk bone marrow aspirates, so we don't really know where these signals come from. So what we did, we performed a computational deconvolution of this RNA-seq data by integrating this with a published healthy single-cell RNA-seq bone marrow niche data sets, which was published last year in Cell. So this is a very comprehensive data set, including a lot of niche cells also, so stroma and endothelial cells from a bone marrow, or from a hip, dissociated hip. And what we then did is that we took the differential expressed genes from our nanostring panels, and we averaged their expression from each healthy bone marrow cell type, and we clustered the genes of the results in a cell-by-gene matrix, as you can see now here on the right side. And we also condensed, like for visualizability purposes, like cells from similar cell types or clusters from similar cell types together. And what we then observe is that we see inflammatory signals actually coming from monocytes, stromal and endothelium. So for example, also, I will not go further into it, but what we heard this morning from Manja Wobes about the alarmins, we also picked this up in our in the monocytes. And then on top, we also see that the T and B cell populations were very affected in MDS versus controls. So we decided to follow this up in some more detail using single-cell RNA-seq. And we were particularly interested in these very rare stromal cells. So we performed flow cytometry or a fax-based enrichment strategy to isolate these very rare cells out of bone marrow aspirates. And we did this for 3 controls, 3 CHIP, and 4 low-risk MDS donors. And we particularly focused on the T cells, the hematopoietic stem cells, and the stroma. At first, I want to share with you our insights about the stroma. So this is the stromal cells we have been able to isolate in healthy CHIP and MDS donors. We are very curious by this interaction because we know that MSCs play an important role in driving hematopoiesis. So at first, we started to identify a population here, the MSCs, that are, let's figure out if we have a pointer. Oh, no, here. The inflammatory MSCs. So we saw in the differential expression analysis that they have a high inflammatory signature, but also they had an increased extracellular matrix secretion. And then we basically curated an inflammatory signature that was really specific to this inflammatory MSCs. And then when we looked at this inflammatory signature across the conditions, and the stroma cell across conditions, we actually already observed that in CHIP there are more inflammatory cells. And we confirmed this with various methods, among which immunofluorescence, and we could see that we actually had CD271, a marker for MSCs, and IL-1 receptor 1 double positive cells already in the CHIP bone marrow, but we found more clonal spots. And then this was further accentuated in MDS, where we saw these inflammatory cells all over the bone marrow. So we observed that the MSCs have a higher inflammatory secretion. And then, of course, since they play such an important role in guiding hematopoiesis, we also looked at the HSC support factors that they secrete. So we also generated a signature for HSPC support and saw this was severely reduced in MDS, but also already in CHIP across the whole stroma. And then when we split it by the different cell types, we saw that the adipoprogenitors that are actually the main source in the bone marrow that are secreting these hematopoietic support factors were, at first, the amount of these adipoprogenitors was very reduced in MDS, but also we see that then in the inflammatory MSCs, they actually have a lower score for this signature. Interestingly, those inflammatory MSCs we found in MDS might have a little bit of a higher HSPC support signature. So also we, for example, here looked specifically at CXL12, an important chemokine for HSC maintenance. And there's a trend towards like a slight higher expression in the IMSCs in MDS. But of course, to highlight, in CHIP, we still have normal adipoprogenitors, for example, available. So we observed that MDS loses its main HSPC support populations and that IMSCs are already present in CHIP. And both in CHIP and MDS have a reduced HP support capacity. Then occurs what is actually driving inflammation in a niche. So for this, we actually had a look at the hematopoietic stem cells. So also here we performed a lot of analysis, but I want to highlight to you that we looked in all the HSCs from controls versus MDS, and then we saw that the MDS HSPCs had an increase in, for example, TNF-alpha signaling, so had a pro-inflammatory response. And then when we performed trajectory analysis on our single cell data, and we sorted our cells basically on the pseudotime from HSCs to progenitors, we looked in the lymphoid and in the erythroid trajectory, and there we saw that already in CHIP and also in MDS, these HSCs on average had a higher secretion of some pro-inflammatory chemokines like CXL8 and IL-1 beta. Next, we were very curious to understand if then the SF3B1 mutant MDS cells, for example, are the ones that are more pro-inflammatory, or if it are the wild types, or what's going on. So here also Max, the PhD student in our group, has developed this computational tool called SpliceUp, sorry for that, which is a computational prediction tool that is based on the cryptic splicing at the 3' end of the downstream or the affected transcripts in 10x single-cell RNA-seq data. And this is also a collaboration with Pedro Mora and Eva Hellström-Lindberg, where they're currently working also on a methods paper for this, and we also combined this with RAGE-seq. So with this, we could identify the mutated SF3B1 mutant cells in our cohort. And then, for example, when we look in the erythroid progenitors, we see that there is differential expression, but in this case, it's quite clear that it has more to do with like translation and ribosomal rRNA metabolism. So possibly this is more reflecting cell stress or for example, higher proliferation in these cells. The wild type cells had a slight more activity in, for example, inflammatory pathways or generally responses to the extracellular signals. But as we also have seen in several talks already during this meeting, maybe RNA actually is not reflecting best what's going on in these mutant versus wild type cells, and we also should have a closer look at protein levels in the future. Yeah, so then as a last single cell population, we looked at the T cells in the bone marrow and actually understand if they could contribute to the inflammatory microenvironment. So we found, very interestingly, 3 distinct T cell populations that were more abundant in already a bit in CHIP and in MDS, which were heat shock expressing T cell population, an inflammatory CD8 population, and cytotoxic CD8 population. And we could also confirm by imaging of our bone marrow biopsies with, for example, here for the cytotoxic cells with granulysin co-staining, that we could identify this increase in cytotoxic T cells in the bone marrow, not only in the peripheral blood system, but actually also within the tissue. And the same for the inflammatory responsive T cells with OLS co-staining. And then when we performed single cell cell-cell communication analysis using a tool called Niches, we also saw that especially the IMSCs and MDS were interacting actively with these inflammatory T cells, and we found several communication angles between the inflammatory MSCs and the interferon responsive T cells that could indicate that they are actively recruited and also actively stay in the bone marrow due to, for example, the fibronectin integrin axis. And then lastly, something that we really hope to also be able to study on a single cell level was the vasculature, but unfortunately it's really challenging or basically impossible to capture endothelial cells from bone marrow aspirates. We were looking into the vasculature. So at first we looked in the angiogenesis capacity of these stromal cells, and we see that if we have angiogenesis, if we look in the angiogenesis signature again, then we see that the adipose progenitors, they have the highest score among all stromal cells we recovered, and they are also often described to be perivascular, and that actually IMSCs relatively have a high angiogenesis score, so indicating that they could induce vascular remodeling. So then we also performed imaging of both sinusoidal and arteriolar structures, and we saw that particularly actually in MDS, we see an increase in the total surface of sinusoids and arteries, which could indicate that this comes due to signals from the inflammatory MSCs close to vasculature. So with this, I come to the summary of our story, and this is currently on bioRxiv and in revision, so if you want to read more about it, you can look on bioRxiv, where we see that compared to the H bone marrow, we have already in CHIP a slight increase in inflammation, maybe driven by already the HSCs themselves, and we see the appearance of heat shock protein expressing T cells and some inflammatory responsive CD8 T cells. We see the first IMSCs appearing that are secreting also inflammatory factors and maybe potentially already start working on the vasculature. And then this is further exacerbated in low-risk MDS, where we see basically we have mainly IMSCs, we see a huge loss of adipose progenitors, and we see vasculature expansion. And then just as a small snippet, we started with a lot of spatial profiling in the group. So we actually aim to further understand how this remodeled niche is occurring, and also is this due to interactions with the mutated cells. So we are profiling healthy, aged CHIP and various leukemic niches, among which low-risk MDS and AML. So we have built multiplex imaging panels to identify immune cells and the stromal populations and also mutated cells. And now we are performing Visium HD, which is spatial transcriptomics methods, where we start to identify diverse niches within the various areas of the bone marrow. And this, this will be the next step, basically, to better understand how this niche is getting remodeled. And this brings me to my acknowledges, where I, of course, want to thank Judith and the rest of the lab, and especially Max, for his great help on the computational side of this project, and all our various collaborators in this project. And thank you for this opportunity.
So thank you also, Dr. Premel. Congratulations again, and it's also quite clear that it is an excellent research program and data. So what is highlighting is that we start with a lot of immunology on Thursday, and we end with a lot of immunology, and I think that is also quite nice. Thank you. I would like to invite all awardees to come to the stage to have a final photo. All awardees, including the MDS Foundation awardee. So please come. Or they are not here still. Yes, we miss one, Dr. Lin. Where's Dr. Lin? He's not there anymore. So, Dr. Steve? Oh, he went. So, would you one photo?
Yes. So it's time for some closing remarks. And I must say that Arjan and I hope that you have enjoyed the MDS Foundation Congress meeting here in Rotterdam. And we certainly did. But we realized that your time has been packed with all the scientific sessions and discussions, and that you may have had little time left to explore the city of Rotterdam. But maybe during your stay here in Rotterdam, you might come across this painting. It's very close to the conference center, and it's made by a Dutch street artist called Daan Botlek, and it's called Make It Happen. So what I would like to do is thank all the people who have made this conference, MDS Conference 2025, happen. First, the MDS Foundation, and especially the Board of Directors. I would like to thank you for choosing the Netherlands as a location for this MDS 2025 meeting. We've been honored to host you here in Rotterdam. And I would like to especially mention Moshe and Leah for all their help in preparing for this conference. Kenneth, who took care of all the logistics of this meeting. And I particularly want to mention Lolita and Netta. I don't know if they're in this room at the moment, but for all your hard work and support before and during this conference. I also would like to thank the members of the local organizing committee here in the Netherlands, all speakers and poster presenters during this meeting, and of course all our sponsors. And if you zoom in more closely to this piece of art, you can see that the tower of people to reach the top is not complete without the active participation of the public. So I would like to thank all of you for your attendance during this conference and for your active participation in all the discussions. And I wish you all a very safe trip home, and I hope to see all of you in Leipzig in 2027. Thank you very much.